Yeah, I would like to give you some insights about the genetic architecture of feed efficiency traits in oil and gas pigs. Just a quick background, so ECO-FCE project aims to contribute to breeding and producing pig and poultry, specifically to improve energy use efficiency and reduce the environmental footprint of livestock production, but also to provide strategies to save resources and costs. A common measurement of feed efficiency is the feed conversion ratio as a ratio of feed intake and weight gain. The breeding goal is to have a lower FCR value, meaning higher efficiency. And the recording and measurement in routine is difficult and costly because you need some feeding stations and have to record these parameters. So we will benefit from the clarification of the genetic background and the development of molecular markers, and furthermore also in strategy for genomic selection. There are several factors driving feed efficiency. I think we more or less covered all of those within this session. Just briefly, extrinsic factors like diet management and climate, intrinsic factors like feed efficiency, digestion and absorption and energy expenditure, and somewhere in between there is the microbiota. And this just argues that there are a lot of genetic factors of diverse biological processes that contribute to feed efficiency. And this is also depicted by the heritability estimates from the literature. So the estimates for feed conversion ratio are moderate, 0.4 in poultry and 0.3 in pigs, and also for the other traits that we investigate, like feeding behavior traits, feed intake and weight gain, they show the low to moderate heritability. The objective of the analysis that we have been done is to characterize the differences in the genetic profile of animals with good and poor feed efficiency, and our specific aim is to identify candidate genes, positional and functional candidate genes that play a role in feed efficiency in both species. This is just a quick overview of the pipeline that we used for the analysis. So we extracted DNA from blood samples, we genotyped these animals on the Illumina platform using the 60K SNP chips. Then the genotypes were called using Genome Studio, as depicted here. Then we followed an imputation approach using fast phase to close gaps within the genotype matrix. And then we combined single marker analysis and multi-marker analysis. And specifically for the multi-marker analysis, we were interested in the contribution of 1 megabase per consecutive windows to the genetic variants. And this integration of both methods were used to identify QTL regions and also to select interesting candidate genes in the background of feed efficiency. For the broiler study, there were an initial set of 5,000 birds from the commercial cock broiler line A, and out of these 5,000 birds, the heaviest 1,000 were selected and tested for feed conversion efficiency between days 39 and 46. 859 birds were genotyped, and the traits that were analyzed are body weight, feed intake, weight gain, and feed conversion ratio. And we are happy to hear that COP is planning some more in-deep phenotyping, which will be, I think, interesting to have those data for the next data sets and studies. So for this data set, the heritability estimates were low. This is on the one hand due to the fact that the broiler line was already under selection for average daily gain and feed conversion ratio for several generations. But on the other hand, the pre-selection also influenced these heritability estimates. So these are two Manhattan plots. On the upper part, it's the single marker approach showing the significance of the markers. So meaning the higher the dot, the higher is significant. And this diagram or this Manhattan plot show the results from the multi-marker approach, and we combine both approaches. So we're looking for the, let's say, most interesting or most prominent SNPs, and then we selected based on these SNPs some QTL regions as indicated here, and within this QTL regions we looked for some interesting candidate genes in the background of feed efficiency. For instance, for feed conversion ratio in broilers, we selected the acylglycerol kinase gene, which is involved in the biosynthesis of signaling lipids, and it's known to be associated with mitochondrial disorders in human. And the second gene that we selected is the GTF2I gene. This is involved in the regulation of growth factor signaling, and it's known that the genomic region is affected by chromosomal rearrangements in humans, and in knockout mice it's known that the gene knockout leads to growth retardation and reduction in body weight. And we selected some coding polymorphism within these two genes and validated the associations, and as you can see, we were able to validate the effects on feed conversion ratio. Furthermore, we used entropy analysis, which are based on the information theory, and they should give the location of the most informative SNPs. And here I would introduce these two posters that are somewhere in the hall, but you can find the exact position on page 63 of the program. So I think the colleagues from Poland who did this study are happy to take some interesting discussions on this approach. For the pig part, we used the Heiling Mexico terminal sialine. The animals were sampled between 2006 and 2015. They were feed efficiency tested during the grower finisher phase, and in total 1,296 boars were genotyped for this data set. And we have different feeding behavior traits, we have feed conversion ratio, carcass composition traits, and also breeding values for this population. The heritability was similar to the literature, so we found moderate heritability of carcass composition traits and also of feed efficiency and breeding behavior traits. And again, the integration of the single and multi-marker analysis. And here I just want to highlight this region on chromosome 11, which points to the DNA GC15 gene, which seems to be quite interesting, especially when we look into the functional annotation. So it's mentioned to be a negative regulator of the mitochondrial respiratory chain, and it restricts the ATP generation. Moreover, we also, or I shortly want to introduce some other candidate genes that we identify. It's again the AQP4, so the aquaporins, which regulate body water balance and mediate water flows. So if you remember, we also found this gene differentially expressed in our expression analysis. So it's quite interesting. So it seems to be quite interesting in the background of feed efficiency. Then there's also the gene, for instance, involved in the regulation of glycolysis, and the PPP3CA gene, which regulates body formation and osteoblast differentiation, were found to be associated with daily feeding rate. This last slide just gave an overview of the identified QTL region. So there were some 26 QTLs identified for the traits, for the analyzed traits in chicken, and around 100 QTLs for the different traits that we analyzed in pigs. And all these QTL regions provide some interesting resource for the development of informative biomarkers and to dig more into the contribution of different genes to feed efficiency. Thanks for your attention.